So anyways, it's good to be here. And yeah, I think there were some points and questions that were advanced by Mark that I think are good. They're legit. And I think as creationists, we need to have good answers to those arguments and challenges. Mark, if it's okay with you, since I did just join, I did catch bits and pieces of the discussion, but can you take some time to reiterate why you believe endogenous
I softened
retroviruses
them up
are?
for you, SFT. Oh, that
Well,
one. Sorry.
I know you guys are best buds. Yeah. So, Mark, take your time. Maybe just go over briefly why you believe.
So I went through the basic ERV sequence of integrase, making a cut, causing long-term and repeats in the edges of it, inserting the or transcribing that sequence into the DNA of the host. Essentially, it is selected at random around the outside of selection areas of where it integrates to. C Science was suggesting, or at least it sounded to me like he was suggesting, that it was specific places where it does have preference for places, but really it's the closest entry point to the genome on the outside of the cell it is infecting. I also explained that it cannot be the case that he suggested they were infecting the body like normal retroviruses do. I said that cannot happen because it has to infect the germline. He was trying to say, why do we only share 205 with chimpanzees? My response was 205. We shouldn't even share one if they are retroviral insertions, because the chances of them being in the same place is so minuscule. 205, we shouldn't even see that. He brought up that they were front-loaded in some way. I don't see any reason to believe they are front-loaded. They have all the hallmarks of retroviruses, and I see no evidence to suggest that is the case. I think that's about it. I'm trying to sum up a large discussion into a very short amount of time.
Okay, Mark, I appreciate that.
Was that okay, C Science? Did I misrepresent you in any way? I wouldn't like to misrepresent people.
Yeah, and well, I think I've got.
I wasn't really listening, brother. I don't know. I was just kind of chatting with Amy.
Okay, well, let's just say it is. If I misrepresented, that's.
Well, did you ever answer the question of why ERVs?
Well, Mark, let me just go back and forth with Mark. Well, there's two Marks, I guess. So, C Science, let me go back and forth with Mark on this topic in order to avoid too many rabbit trails there. So, essentially,
go for it.
You would argue, Mark, that the 205 full ERVs, as you pointed out, LTR, GAG, POL, and ENV components of the ERV. Actually, Amy, if you don't mind, I've got a diagram of what Mark's talking about with the ERV on screen. If you could, I think that'd be helpful for all of us. Okay.
Ooh, infinite regress.
There we go. So yeah, that brief moment. So as Mark was saying, we've got your classic ERV. We got different classes of retrotransposons or retro elements. ERV is one of them. And so you can see the structure, as Mark has pointed out, LTR, GAG, POL, and ENV, or envelope protein. So this same structure is what we see with retroviruses as well. Exogenous retroviruses like your HIV, which would infect from outside. Endogenous meaning occurs from within. And so that sequence similarity would suggest to the evolutionist that these were ultimately derived from ancient exogenous retroviral infection. So, Mark, you would say those 205 plus you've got thousands of solo LTRs, right? That basically lack your key endogenous retroviral components. Those being shared between.
I just want to point out really quickly, and exogenous from the outside would infect in different places, and they have done tests to show that is the case.
Well, I agree that
you agree with
external viral infection occurs for sure. And we find nested hierarchical patterns in the distribution. I do remember you pointing out that we share more with the chimpanzee than we do with the gorilla or the orangutan or the old world, new world monkeys. So I think the question is, if these are actually viral infections, how can a creationist explain independently 200 plus endogenous retroviral elements integrating into the exact same genetic loci in humans and chimps if they were not inherited from a common ancestor, basically?
Yes.
Okay.
Mhm.
Yeah. And so the question that I typically ask, and it's definitely not new to you, Mark, is are these really the ancient remnants of past viral infections, or are they mostly created units of DNA function? If they are created units of DNA function, at least the fixed ones, okay, we can get into that, then the reason why they are shared, let's say the 205 plus the thousands of solo LTRs that we find, is for common design reasons. The same reason all modes of transportation share an engine in common, if they are created units of DNA function. Now, I would argue that we do have examples of true endogenous retroviruses, but those would generally occur at a low frequency. For example, I typically cite a paper where they examine HERVs, human endogenous retroviruses, and there's a subset of the human population that has a very specific HERV. And so I think we can safely conclude, yeah, that is the result of a true endogenization event, where basically an exogenous retrovirus integrated into the germ cell lines, egg or sperm, and then managed to be passed on to become endogenous. So I do believe that that happens, but I would argue the majority are reflective of initial design. And one reason I'd like you to speak to this, Mark, and take as long as you need. One reason why your endogenous retroviruses share that sequence similarity with exogenous retroviruses, the GAG, the POL, the ENV, the LTRs, is because amazingly, one of their functional roles involves tumor suppression. And they do this through viral mimicry. And so basically, transcribed ERVs, they give the cell the appearance of being invaded by an external and harmful retrovirus. And as a result, by the way, this is stimulated by the P53 protein, the guardian of the genome, basically. The immune system targets that cell with more destruction, which as a result destroys those tumor cells. But this functional role can only be carried out if the endogenous retrovirus looks like the exogenous retrovirus that it's mimicking. And so it requires the GAG, the POL, the ENV, and the LTRs to perform that functional role. Go ahead, Mark.
Okay. So when you say sort of, so I did get the exogenous, and I really want people to understand this. If an exogenous retrovirus, and that's one coming from outside that hasn't been infected the germline and become endogenous or a part of the normal genetic makeup, if that infects, it will be in all different parts of the genome in different cells. It will be in different parts. So I just want to make that clear. What is your mechanism? And I want to ask this sort of hypothetically. So if you've got these endogenous retroviruses, they have the same pattern as what you call the ones originating from a created design kind of thing. It seems to me there is no way to tell the difference between them. And I'll point you to, I think we've discussed it before, Phoenix Rising, where they basically got an endogenous retroviral sequence that was inert, and they reactivated it, turning it back into the virus. Because yes, some of these have been co-opted to be beneficial. I can get papers showing that they've been co-opted, but some of them do nothing at all. Or there are retroviral elements which are devastating should a mutation return them to working order. So it seems to me like, Donny, you don't have any way to distinguish between the three, except for, and I'm sorry to say this, what's sort of convenient for what you're saying, that the ones that do something good are not the retroviruses, they're created, and the ones that do something bad are the retroviruses. And, you know, you would understand why that wouldn't be a good solution to determine what has been created and what hasn't been created. You mentioned the P53 viral element, and I agree. If that's knocked out, it's absolute devastation, because it does do exactly what you say it does. And I'm not sure if you're saying sort of it mimics the ability of retroviruses to suppress other infections. I think it's described by scientists as just that is what it's doing because it is a retroviral element. And we've seen retroviruses do that before. That is what they do. In order to stop other things from infecting the host they have infected, they suppress other infections. So I don't know why we'd sort of appeal to a mimicry of the behavior when they might just be doing that behavior. And sort of you also have these other retroviral elements that, or, you know, if they are created things like you say, that we do knockout tests and knock out the genes and they do absolutely nothing, and we know they do nothing, like Phoenix Rising, which we can turn back into a virus. But my question to you is, what is your methodology for determining the difference between viral elements and these created segments, and how do you put that into a scientific lab to determine what's what?
Excellent question, Mark. I appreciate you asking it. So you brought up a few points and I wrote them down as quickly as I could. And so your main question is, how can we tell the difference between a created ERV, a created unit of DNA function, and a true endogenous retrovirus, something that's actually the result of, and you, Mark, you explained it well, that essentially when an exogenous retrovirus infiltrates and infects a reproductive cell, if it's passed on, that the progeny has that viral DNA in every single one of its cells. Now, how can we tell the difference between that which is created and that which might be the result of a true infection that became endogenized? I would argue it comes down to fixed versus unfixed ERVs. And so the majority of your ERVs that we discuss, such as the 205 full ERV components, as in the ERVs that are not just solo LTRs, but actually have your GAG, your POL, your ENV components, okay, your LTRs on either side, those are fixed. What that means is they are stuck in place. Every single human being has them, and all your chimpanzees have them, which is why the evolutionists would even argue we share them in the first place, right? They'd have to be fixed. Then you have what's called unfixed ERVs. What that means is only a subpopulation has them. Okay, so in humans, those HERVs, human endogenous retroviruses, there would only be a small frequency of people that have them, maybe 5%. There might be a higher frequency where 80% or 90% of people have them, but not 100%. And so how I would determine that which is fixed versus, or that which is created versus not created, is whether or not they're fixed. So if every single human being has them and the chimpanzees share in similar genetic loci those ERVs, then I would say that those are most likely created. If they're unfixed and only occur in a low frequency, let's say within humans or chimpanzees, they are the result of a true infection. Before you respond, I do want to make a point about the Phoenix virus experiment, because that's important and that's a good point. But the debate is not about whether or not internal endogenous retroviral elements can produce viruses, harmful viruses that can basically jump from one species to the next. Because the escapee hypothesis that we hold to, that argues retroviruses originated from within the genomes of their hosts, right? Because the question that I like to ask is, what came first, the host or the retrovirus? Because the retrovirus requires a host to replicate. And even many in the conventional literature, they would hold to what I'm saying, the escapee hypothesis, that basically your retroviruses came from within the genomes of their hosts, and then they've crossed species and have become infectious. So that's an agnostic line of evidence, because all the Phoenix virus experiment did was resurrected a dead virus from a host genome. Well, we already agree as creationists, at least I do, because I hold to the escapee hypothesis, that that can happen. And so it's going to come down to your fixed versus unfixed ERVs, but also function. So I would predict that those fixed ERVs are going to have functional roles in the immune system, cell stress responses, determining cell types, gene regulation, gene expression, placental development, for example. And so that's the best way I'd answer your question, Mark. Go ahead, speak to that.
Yeah, I don't really see fixed versus unfixed as being particularly definitive on how to determine that these have some sort of function or not, because we've done knockout tests on fixed retroviral sequences and they don't do anything. The problem comes that it's sort of a post hoc kind of rationalization that you can say, hey, the fixed ones are there. I mean, we would agree that the fixed ones wouldn't be that detrimental because they have to be, but we know that if the fixed ones mutate, they can be cause genetic problems. Would you agree to that?
Good question, Mark. I don't believe it's post hoc because, well, firstly, to answer your question, yeah, there are a lot of endogenous retroviruses that can be hit with deleterious mutations, and that could lead to multiple sclerosis, for example. There's a lot of work done, right? Different, all types of diseases, right? That is essentially degeneration of a unit of DNA that wasn't originally involved in those kinds of diseases. But mutations can take a functioning, healthy DNA unit and alter it in a way where it causes disease. So I don't think that's an issue. But the nature of the fixed ERVs, it's not that every single ERV is going to have a role that is detrimental to the organism, right? So, for example, we've learned that mutations are not necessarily completely random. A lot of them are non-random because mutations oftentimes will target a spot of the genome that might not be involved in something dealing with the heart or something dealing with embryological development. Because if a mutation hits a specific section of the DNA involved with something as critical as the heart, well, the organism's going to die. And so typically those parts of the genome will be preserved, but mutations will more frequently hit regions of the genome that might involve redundant areas or even areas involving the immune system, because we have a lot of DNA elements that assist in the immune system. And so we could probably afford a few DNA elements being hit with mutations, but we still got a lot of backup code essentially. Does that make sense to you, Mark? And
yeah, it kind of, though, it seems like you are designating these retro, or what appears to be retroviral elements, or the created sequences that you're talking about, by their usefulness and functionality rather than by any kind of physical evaluation of them at all.
Well, I would argue if they're fixed, meaning every single human being has them, but they're also involved in some kind of function like tumor suppression, which involves viral mimicry. I mean, the endogenous retrovirus has to look like the virus that it is mimicking in order to perform its job. Like if an FBI agent was trying to infiltrate the mob, right? He has to look the part. He's got to look like the mob. You know, we've all seen those movies, The Departed, Goodfellas, so on and so forth. But if he showed up with his FBI badge and in his uniform, well, he's not going to get too far.
I'd go with the Terminator on that one, actually. Yeah. Like, you're a robot. You're here to kill everybody. You wrap yourself in the flesh so that none of the detectors can find you.
Yeah, I mean, you could say that a robot like Terminator has to look the part, so it's not, caught in its disguise. So a lot of these ERVs, they function in fighting off viral infections. They function in the immune system, and they do so by looking like the enemy, by looking like that which they fight. And so, yeah, if these are fixed, all 205, let's say, that all humans have, and they share with the chimpanzee, I would argue that those are the created units of DNA function. But there are many ERVs that occur at a low frequency. They're unfixed, meaning not every single person has them. And so that would tell me, okay, they're probably damaging somehow. They're most likely disease causing, right? Maybe they result in the multiple sclerosis or types of autoimmune diseases. They're probably not functional, especially in something like embryological development. And that's partly why they're unfixed, because they were the result of an exogenous retroviral infection in the past. It became endogenized, but it only spread through a small part of the population. And we do have examples of that in humans. Does that make sense, Mark?
Not really. I just don't know why, when creating a sort of a sequence in order to help us with something, that you would basically make it exactly like a retroviral sequence, including the LTRs, and then make some of them functional and then some of them non-functional, and sort of not activate them at all. That doesn't seem the more parsimonious explanation does to be that they're simply the ancient remains of viruses, some of which have been co-opted, as we know that any part of the genome can mutate and have these become beneficial, especially in disease prevention, because of the way that retroviruses behave, even retroviruses like AIDS that are currently basically preventing infection because they want the host, and have been a result of common ancestry. I don't see why something would design it in such a way that there is so much, as through knockout tests, so much neutral area, and that the ones that do something useful for all intents and purposes seem to be the ancient remnants of those retroviruses with exactly the same structure, exactly the same marks left by their integration, and sort of say, well, that seems like the most probable explanation. To me, it seems like a very far-fetched explanation.
I appreciate those points. I would argue that the most parsimonious explanation is the created units of DNA function model, precisely because I've not seen a convincing response. And I've got a specific paper in front of me. Amy could either screen share or not a big deal, but in case people wanted to see it. And it's a paper from 2021. It's called Switching Sides, How Endogenous Retroviruses Protect Us from Viral Infection. It goes into, the paper is a fascinating read. It goes into each component, the ENV, the GAG, the POL. Okay, it goes into the details on how each component of the ERV is actually necessary and required for the ERV's function in fighting off infections. And so I see it as front-loaded antiviral programs. And that screams of good design. I mean, our computers would crash after a long time without any kind of antiviral protection built in or added in as well. But these functional roles, one specifically being the role in embryological development. There's several papers that essentially point out, if we didn't have these endogenous retroviruses in our systems, mammals wouldn't exist, as in we literally require them to exist. And so my challenge to the evolutionist is, show me where an exogenous retrovirus has infected, like HIV, that's an exogenous retrovirus. Where have they ever infected in the right way, which is in the reproductive cells, has been passed on, and then through mutations over time, basically the harmful parts of the endogenous retrovirus are destroyed. And then through either co-option or beneficial mutation accumulation, that ERV evolves something that is necessary for placental or embryological development. You know, my challenge is to the evolutionists is to show us how that's possible, how that's happened in the past, and if we've observed it today, either in nature or the lab. Go ahead.
Yeah, so this switching sides, it absolutely disagrees with you and says that they were co-opted retroviruses, and they've outlined all of the viral infection and simulation that they've done to show that they were retroviruses. So this paper that you've said, and I've actually got it in my list of papers to support the endogenous retroviral evolution model, because it 100% supports it. You know, so sort of they've been identified as pre-existing viral matter. So this paper doesn't doesn't help your case because they've explained how it has been co-opted. So yeah, this whole idea that they have been included somehow, I don't know how this paper helps you. What was the other thing you asked? You asked for where a retroviral element has infected humans and what? Sorry, I do apologize. I was kind of lost in the paper.
No, that's okay. I'll reiterate it and then I'll respond to your point thereafter when you're done with your arguments. Essentially, I pointed out how without endogenous retroviruses, the placenta being critical to pregnancy and embryological development. Right. Have we seen that kind of?
So it's not critical to pregnancy. It's not because we've got animals that are very similar to mammals, yet they give birth without sort of a placenta. So we've got the marsupials that do not have that retroviral element in it, that do use a pouch system and an embryonic sac to instead they don't have a placenta. And that's kangaroos and koalas and very similar animals to the ones we have today. We have the monotremes, which don't even have a sac, and they're further on. They don't have the retroviral elements that produce live young. They have a shell system still, because that did not split off. So when you say it's necessary for pregnancy, in mammals, yes, because we all have split off with that retroviral element, but there are other animals without that retroviral element. And they're very similar to mammals, but they split off just before that developed. So I can show that that's completely incorrect. We do not need that retrovirus to become pregnant, only to become pregnant as placental mammals, and that's all.
Okay, I appreciate those points. Those are good points for me to address, and so I'll do them in turn. So first, you pointed out how the paper claims that these functional roles involved in the immune system were co-opted, but they don't actually provide any evidence for this co-option. They don't show us how or when or where a pre-existing endogenous retrovirus gained a novel function in the immune system, or determining cell types, or in tumor suppression with their role in viral mimicry. They're basically just asserting it without evidence. So assertions without evidence isn't a real good line of argumentation. So you need to see or provide evidence for these novel functions coming about. But I find it funny because the evolutionists oftentimes ask, well, why do the ERVs look like retroviruses? Why are they built the same way? Why do they have the LTRs, the GAG, the POL, the ENV? And it's like, we point out as informed creationists, well, they require their sequence similarity to retroviruses to literally carry out their various functional roles. But it's not good enough. And it makes me wonder if the evolutionist really is looking for an answer, because that is an answer. They require those similarities in order to basically do their job. And so the next point you made was, and I do mean this in the nicest way possible, but I feel like you completely dodged it, Mark, because again, you didn't show us anywhere or provide any technical paper that has actually demonstrated an exogenous retrovirus infecting in the reproductive cell lines, whether egg or sperm, being passed on, mutations then wipe away over time, because this is in theory how it's supposed to take place. Mutations wipe away the harmful aspects of the endogenous retrovirus, and then eventually through co-option or beneficial mutation accumulation, it evolves the ability to function in embryological development. I don't want evolutionary philosophy. I want to actually see in real time, empirically, how this is possible. You pointed to different modes of reproduction. Animals without a placenta, some mammals with a placenta. Okay, that's not telling us how the endogenous retroviruses evolved the ability to function in the placenta. Yeah, we have a variety of designs in the biological world. I mean, just look at engines and modes of transportation. You've got a diesel, you can get an electric car, you can get a regular engine, you can get a hybrid. Okay, yeah, there's a diversity in engines, but just saying that, well, a diesel engine exists, and so that means, you know, an electric car or a hybrid car, it's possible to not have a diesel or something like that. No, you're not telling us how the placental development came about. yes, we see variation in the biological world. It's not really a surprise, but I want to see how an endogenous retroviral element can actually go from something that's non-functioning to something functioning in embryological development. And that is my challenge. Go ahead.
Okay, so I want to share my screen. I put the articles in here. So the problem here is that it's not claiming anything. That's its conclusion. So I think sort of saying, well, it claims this is a bit misleading, to be honest with you, because it's the conclusions of the paper that you're citing, Donny. So if your paper doesn't come to the conclusions that you want, change what paper you're referencing. This is a paper that is in their citations, I might add, so you probably should have backtracked the citations, that the HIV-1 infection leads to increased transcription of human endogenous retrovirus, HERVK, proviruses in vivo, but not to increase viron production. So it was not replicating, yet it protected against proviruses. There's another one that I have that they also referenced. So this is the reference they provided because they're not just sort of saying this out of the blue, but in that paper, they will make citations to the papers that they're building off. And that's what you've got to go see, because they will cite these papers saying, hey, we know that they are co-opted from these papers, and that is what's providing us the thing. I've shared the other one. Anyway, this is TRIM28 mediates primer binding site-targeted silencing of marine leukemia virus in embryonic cells. So that's one affecting, as you asked for,
can you share
from
your
the
screen,
embryo.
Mark?
I thought I was. Sorry, I may not have been.
No worries.
Yeah,
I think Amy or James from your end.
It never came up on the.
Yeah. I put them in the chat as well. Okay. So if you do need a reference, they're in the chat as well.
Okay, I pulled them up. Keep going, Mark. Apologies.
Yeah. So when you're looking for we've got to know the exact mechanisms, exactly how this occurred in placental development, we might not be able to show that. I mean, we can show the endogenous retrovirus that's led up to placental development, and we've got tons of papers on them that it was a retroviral element that was co-opted in order to provide this thing. But you're sort of saying, hey, I want to see the virus before it was co-opted, and I'm not sure we can show you that. Too much water under the bridge. It's been changed way too much. It's been mutated way too much. So I think that's an unrealistic expectation. But what we can show is that these viral elements do infect things and they do get co-opted. So there's no reason to think that it was the creation of some design. Instead, it is what it appears to be, which is the co-option of a retroviral element that has been inserted at the germline.
I appreciate those responses and papers, Mark. I do hear a lot of background noise or echo coming. James, I wonder if it's from you. Because okay.
Yeah, everyone else is muted, yeah.
Amy, if you could, or James, share my screen as well, because I did pull up those papers that Mark has cited. And so I see that, Mark, as an admittance, and I appreciate it. An admittance into the fact that, yes, these co-option events, the evolution of these novel functions, by the way, that the conventional literature admits, if we didn't have these elements with these functions, we literally wouldn't exist. And so when an evolutionist asks an intelligent design proponent, why do we share these elements with the chimpanzee? I feel like they're not necessarily steel manning the position or understanding the design hypothesis. Because if we have DNA elements that are literally necessary for biological life to exist and function in the first place, that's like asking, why do GMs and Chryslers share engines? I mean, you can get rid of a lot of parts on a car and the car is still going to function and run. But the second you get rid of the engine, chances are that car is not going to go too far. So many ERVs function in cell processes that if they're hit with a mutation, it's not necessarily going to kill the organism. There's a lot of backup code, a lot of backup genetic elements. But an element like some of these ERVs that function in the placenta, yeah, that's necessary in order for life to survive or exist in the first place. And so I've got a whole presentation on that paper, the original paper here that we're talking about, switching sides. And as you can see here, they go into how the LTRs act as enhancers and increase antiviral gene expression. They go into the functions of the ENV proteins and how they're associated with these immune system related functions, the GAG proteins as well, right here, besides ENV, endogenous retrovirus GAG proteins can also restrict retroviral replication. And so everything about the ERV is necessary, is my point. And so it's no surprise that they look like the retroviruses that they're protecting us against. So here's the papers from Mark. And I'm just looking over them.
So if you don't mind, Donny, you can actually take as long as you want to finish up your point. But then after you have a point and Mark does, I'm actually going to hand it back over to you, Donny, for your for everyone's outros. But you've sounded like you were just finishing up a point. Take all the time you need.
I appreciate that, Amy. You're a great host and you're a very fair moderator. So that is much appreciated. And Mark is always an excellent interlocutor. Seriously, when Mark joins the open mics, I think to myself, here we go. This is going to be a good show, because that's the whole reason why we do these discussions, is so we can have some interesting back and forth. Exactly. And that goes for passionate back and forth. So I'm going to read these papers, of course, on my own. But I would just appreciate if Mark showed me where in these papers we are seeing empirical or real-time evidence or even a direct answer to my challenge. You know, like here it says
your challenge was specifically about placental.
Sorry, these papers showing how the immune system roles came about?
No, you said that like you wanted evidence for placental, the retrovirus that sort of allowed us to have placental development.
Well, you can go down the line. ERVs function in determining cell types. They function in cell stress responses. Gene expression, that's basically the process by which a gene gets turned on in a cell to make RNA and proteins. And guess what? ERVs are involved in that. Gene regulation, the regulation of the process known as gene expression. So any of these functional roles, show me how any endogenous retrovirus evolved this functional role. And if an evolutionist can't show me that, it's evolutionary philosophy. It's not actually science. It's just an assertion and a claim. And these papers can conclude all sorts of things. But if those conclusions are not backed by empirical scientific data, then it's a conclusion not based on science. It's mostly based on story.
Well, science is inference, and that's what we can infer from having these retroviral elements co-opted. So there's no reason to think they were created if they can be co-opted. And sort of the whole idea of
where does it show them being co-opted?
Hold on a sec, Donny. The whole idea of we wouldn't exist, which is what you said, we would exist, we just wouldn't give birth by placental means. We would probably give birth by monotremes, like we would lay eggs, or we would have a sac, an embryonic sac like kangaroos. The whole idea that you're sort of saying, hey, but we wouldn't exist, yeah, not exactly as we are now, but that's sort of there's no counterfactual to that. Because we did evolve this ability, you can't say, well, we wouldn't exist, because we would. Because there are animals that do exist and give birth. And you said it again, that they're required for pregnancy. No, they're not required for pregnancy. And the fact that there are animals without these ERV sequences that do give birth, whether through embryonic sac or egg, shows that it is not essential for life, as you sort of. So you said directly, in order for life to survive, and that is not true, because there are. I'm not sure what you're typing out. Do you want to maybe? in order for life to survive. No, there's life forms without these ERV sequences as you go further and further back. And this is the nested hierarchy that we're talking about. The further away from us on the evolutionary tree you get, the less ERV sequences are shared between life forms. So you're saying it's essential to survive, but other things survive without them. We just may be more optimal because we have evolved and these mutations have actually been beneficial. Again, you've quoted the paper Switching Sides. The entire point of Switching Sides is that once damaging and detrimental viruses can be co-opted to be beneficial, and that's exactly what they've pointed out. I've given examples where viruses are co-opted by mutation to be beneficial to their host, and those are the citations they use to show that it can happen. And as a product of inference, they have made this inference quite tentatively. I think that they're not exactly sort of saying, hey, we've figured it all out. But they've certainly said, hey, there is no reason to think these were created when they look like endogenous retroviruses, they act like endogenous retroviruses, but they have been co-opted, like we have seen time and time again, to be beneficial for the organism. Again, you mentioned that it's necessary. No, they're not necessary. Other life forms exist without these viruses. The whole thing, what it seems to me, is you're sort of saying, well, if it's beneficial, it was created. And there's no reason to think that whatsoever. So as you go further and further back, you've got to explain how, if it is so necessary, how life forms actually exist without these ERVs.
And Donny is going to be able to answer because he is going to get his outro. We all are going to get our outros. He was just the latest to join us, which means he's the first to go, and we're going in the opposite order we came in. We have been skeptic and ranting on why evolution is not true, and we are going to our outros. Woohoo! With that, we're going to hand it off to Donny. Thank you so much, SFT, for joining us tonight on Skeptic and Ranting. But we have very specifically have been talking about why evolution is not true. Dun, dun, dun. I want to thank you. What do you have got going on in the interwebs? And what are your final thoughts on the subject as a whole?
Appreciate it, Amy. This has been a lot of fun, and we definitely need to do more of this. I love discussing this topic, and I especially love discussing this topic with informed interlocutors. Makes it a lot more fun and engaging. And so my last points, based on what Mark said, to me, it's like saying, well, an electric car requires an electric engine to run properly. But it doesn't really require an electric engine to run properly because we've got cars that run on a diesel engine or a regular engine or hybrid or whatever. Well, what we see in the biological world are some creatures that reproduce or exist with a placenta and some that don't. So you can't point to the ones that don't, and therefore, well, the placenta, in these mammals must have evolved. That's just an assertion. That's just evolutionary philosophy. We need supportive evidence for that, and that is still my challenge. That I feel like after years and years of engaging the topic of ERVs, we've yet to see this co-option that is often claimed. We've yet to see these pre-existing endogenous retroviruses going from non-functional to something functional in embryological development, functioning in the immune system, cell stress responses. Again, we talked about their role in tumor suppression. Literally, they could not carry out that role if they did not look like the enemy, if they did not have those sequence similarities to exogenous retroviruses. But this all comes down to, or back to, and here's the last thing I'll say, the bigger question, which is what came first, the retrovirus or the host? Because retroviruses require a host to replicate. And so with the Phoenix virus experiment that Mark talked about, it's agnostic, because I already agree that viruses can come from host genomes and can originate from pre-existing ERVs. And so if the escapee hypothesis is true, then we expect all of this data. So I appreciate. Actually, last thing I'll say: nested hierarchies. Yes, if these are created units of DNA function, Mark pointed to nested hierarchies, then yes, it's no surprise that we would share more with the chimpanzee than we do with an orangutan or a dog or a fish, because we already share more in terms of morphology and anatomy and physiology with a chimpanzee than we do with something like a dog. And that's why it's all going to come back to that important question: are these really the ancient remnants of past viral infections, or are they created units of DNA function? How can we answer the question of what is created and what's not? I point back to fixed versus unfixed ERVs. And this was a lot of fun. Amy, I appreciate it. Thanks.
Yeah, so I always find the sort of engine analysis really weird and strange, because there's no fixed anything in engines. Like, you can't fix anything because there is no propagation of those genes, because engines don't have genes. It is a weird argument, because when you're talking about mutations, you're talking about endogenization of retroviruses. There is no mechanism for passing DNA, because there is no DNA. It's the weirdest thing. I don't understand it. I really don't. You cannot make an analogy between electric engines and human DNA. It's just not possible. I don't understand it. Nested hierarchy, yeah, and we see, like, not only do we see these things with benefits, we see also the same remnants of retroviral elements that do nothing whatsoever. So I think in order to even approach that they were created, you've got to explain why they were created to do absolutely nothing. And sort of, we can do knockout tests and they have no functionality. So you've got to explain that somehow. I think that Ember was absolutely right. It's an argument from incredulity. I don't see how evolution could work. So we've got to come up with these ideas of, well, even though we see retroviral elements doing this, we see them inserting like this, we see them making the same patterns, we see them providing immune benefits, like the HIV virus versus HMLT2, I think it is, or one of the HERVK viral elements. It provides sort of a resistance to it. That has to be a created element rather than everything we see in front of me. I think that has no reasonable evidence for it, whereas the other one, we have a ton of evidence suggesting it's the case from all of the things that I've been through. So sort of asking for this specific case of placental development when saying, hey, life wouldn't be possible without this placental. No, it definitely would be possible. It just, humans wouldn't be here. But that's sort of taking assuming the consequent kind of thing, or sort of, sort of saying, hey, if humans didn't have this thing, then we wouldn't be here, therefore it was created. No, that none of the evidence points that way. So I mean, Donny can bring up studies. They do disagree with him. I agree with the authors of the studies that they have been co-opted. We've seen co-option happen. If you do check the citations of that paper and, check it out, yes, that's exactly what they're saying. So I'm going to go with the people who wrote the paper rather than, unfortunately, some people have a religious interest in it not being true. And that's unfortunate because they don't have a lot of evidence for that apart from their incredulity that it could happen naturally. And that isn't an argument. Thank you.